Hello, everyone. My name is Zhou Dayong from National Engineering Research Center of Seafood, Dalian Polytechnic University. I'm glad to give a presentation here. Today, my topic is oxidation kinetics of polyunsaturated fatty acids being esterified into glycerophosphocholine and glycerophosphoethanolamine in dry scallop during storage. My presentation includes three parts. Firstly, let me introduce the background for my research briefly. Surface, including bivalves and univalve, is one of important seafood. According to Food and Agriculture Organization statistics, the total production of marine surface was about 19 million tons, with an output value of 20 billion US. Many surfaces, such as oyster, clam, mussel, and scallop, all have huge production and economic value. Except for other delicacies that favored by consumers, surfaces also serve as a rich source of bioactive components, including proteins, peptides, minerals, vitamins, taurine, and omega-3 polyunsaturated fatty acids. Therefore, surface can be regarded as a healthy food. So far, more than 20 species of marine surface from Bohai Sea and Yellow Sea of China have been studied by us, and they contain 5 to 15% of lipid in dried bases. Our studies indicated that the lipids in marine surface contained a high proportion of PUFA. Take scallop as an example. For different body parts, PUFA account for about 50% of total fatty acids. Among PUFA, EPA and DHA are dominant, and the two fatty acids account for about 40% of total fatty acids. Therefore, surface may be an important source for dietary omega-3 polyunsaturated fatty acid. The surface lipids are rich in polar lipids, especially phospholipid. Take scallop as an example. The lipids from different body parts were composed of triglycerol, monoglycerol, diacylglycerol, phospholipid, free fatty acid, and cholesterol, among which phospholipid and triglycerol are predominant. Actually, for most of the surface species we studied, phospholipid normally account for more than 50% of total lipids. The surface phospholipids are also composed of different phospholipid classes. Take scallop as an example. The phospholipids from different body parts are composed of PC, PE, PS, PI, PZ, lysopC, and SM. Among the phospholipid classes, PC and PE were dominant, which account for about 40 and 20% of total phospholipid, respectively. Surface is an important dietary source for omega-3 polyunsaturated fatty acids. However, as the moisture-rich organism, surface is prone to rapid microbial spoilage after harvest, and the nutritional components tend to degrade while complex biochemical reactions, which lead to a significant loss of quality, shorter shelf life, and lower commercial value. To overcome this problem, various processing and preservation methods, including freezing, drying, freezing, seasoning, canning, are utilized to extend the shelf life of surface. Some processed surfaces, such as dried, fried, and canned products, can be preserved at room temperature because they are not prone to microbial corruption. However, the presence of high amounts of PUFA in surface make them prone to oxidation, which will impact the edible quality and nutritional value. The PUFA in surface may exist in different forms, such as trace glycerol, phospholipid, and free fatty acid. It has been reported that the PUFA in different lipid classes in different food systems have different oxidative susceptibilities. It's widely accepted that fatty acids in free fatty acid form are more susceptible to oxidation than those in trace glycerol form, due to their greater capacity to catch trace metals. However, there have been inconsistent results with regard to the oxidative susceptibility of trace glycerol and phospholipid. Therefore, in this presentation, take dried scallop as a representative surface. I will introduce our latest research results about the difference in oxidative susceptibility of the same PUFA in different lipid classes and phospholipid classes. The second part, results and the discussion. First, the oxidation kinetics of trace glycerol and phospholipid in dried seafood and the effects of the polyphenolic antioxidant were investigated. The scallop adult mussels were marinated in antioxidants of bamboo leaves (AOB) and freeze-dried. The dried samples with various treatments were subjected to accelerated storage, and the lipid fractions containing trace glycerol and phospholipid were ejected from dried scallops at a selected storage time. The change in content of 4 representative PUFAs being esterified into trace glycerol and phospholipid were fitted with oxidation kinetic models to obtain the rate constants that can reflect the oxidative susceptibility of different lipid classes. Using silica gel column chromatography, trace glycerol and phospholipid fractions were separated from the total lipids ejected from dried scallop adult module. The high-performance thin-layer chromatography imaging showed only one spot for each of the trace glycerol and phospholipid fractions, and the thin-layer chromatogram also showed a distinct peak for each fraction. This indicates that the two lipid fractions have high purity. The fatty acid composition in trace glycerol and phospholipid fractions purified from scallop adult mussel were analyzed by gas chromatography. ALA, AA, EPA, or DHA were found to be the main PUFAs among all fatty acids. Thereby, the 4 PUFAs were selected as representative in the investigation of oxidative susceptibility of different lipid classes. The results indicated that the contents of the 4 representative PUFAs being esterified into trace glycerol and phospholipid all decreased with increasing storage time and 25 degrees centigrade. A similar changing trend was found when the sample was stored under 15 degrees centigrade. The contents of ALA, AA, EPA, and DHA being esterified into different lipid classes in dried adult mussels stored at various times were fitted with the zero-order and first-order kinetic models. The results show that the change of the 4 PUFAs in any lipid formed follow the first-order kinetic model better, indicating that the oxidation rate is directly dependent of PUFA concentration, but is independent of oxygen concentration. This is likely due to the presence of high oxygen concentration in the storage environment and the limited lipid content in dried adult model. Therefore, the reaction rate content obtained from the models can be used to determine their oxidation rates. As shown in the figure, the oxidation rate content of PUFAs in triglycerol form was significantly higher than those of PUFAs in phospholipid form, indicating the oxidation rate of phospholipid is slower than that of triglycerol. The main PUFAs, especially for EPA and DHA, in seafood, are located in the SN2 position of phospholipid. It was speculated that the tightly packaged molecular conformation of phospholipid can hinder free radicals and oxygen to attack the PUFAs at this position, thereby can inhibit the formation of hydroperoxide. Meanwhile, our results show that AOB significantly decreased the oxidation rates of ALA, AA, EPA, and DHA, being esterified into the two types of lipid classes. AOB is a polar natural polyphenolic antioxidant that can be used in food, as permitted by different governmental bodies, including the National Health Commission of the People's Republic of China. It contains more than 40% of total phenols, according to the manufacturer's certificate of analysis. Phenolic compounds in AOB have high content of hydroxy group, a group that can donate hydrogen atom to scavenge free radicals produced during lipid autooxidation, transforming them into quinone structures. As a consequence, the chain reaction of lipid autooxidation is broken, and the degradation of PUFAs is inhibited, causing the decrease of lipid oxidation rates of AA, ALA, EPA, and DHA in any lipid forms. We further calculated oxidation inhibition rate through the equation, which reflects the inhibitory effects of AOB on oxidation kinetics of PUFAs being esterified into different lipid classes. The results show that the oxidation inhibition rate values of the 4 PUFAs being esterified into triglycerol were significantly lower than those of phospholipid, indicating that AOB can more effectively inhibit the oxidation of phospholipid compared with that of triglycerol. This is maybe because the hydrophilic antioxidants should more closely participate at the polar lipid and locate at the site of oxidation reactions. Secondly, the oxidation kinetics of PC and PE in dried seafood and the effects of the polyphenolic antioxidant were investigated according to a similar procedure as presented above. Two phospholipid fractions, including PC and P, were separated by using preparative thin layer chromatography. Their purities were confirmed by direct infused tandem mass method using triple quadruple mass spectrometry in positive I pattern through selective detection. The direct infusion tandem mass spectra of the purified fractions just showed the distinct MS signals of GPC or GPE, not the signals of other phospholipid classes, indicating that the phospholipid fractions have high purity. The results indicated that the content of 4 representative PUFAs, including ALA, AA, EPA, or DHA, being esterified into PC and P are decreased with increasing storage time at both temperature. The content of ALA, AA, EPA, and DHA being esterified into different phospholipid classes in dried adult modules stored at various times were also fitted with the first-order kinetic model better. The oxidation rate content of PUFAs in PE form was significantly higher than those of PUFAs in PC form, indicating the oxidation rate of PC is slower than that of PE. Different phospholipid classes have different polar hand groups, which may result in different oxidative susceptibilities. This could be due to the fact that they are exposed to different pro-oxidation environments caused by their different polar hand groups. Meanwhile, our results show that AOB also significantly decreased the oxidation rates of ALA, AA, EPA, and DHA, being esterified into the two types of phospholipid classes. The oxidation inhibition rate values of the 4 PUFAs being esterified into PC was significantly higher than those of PE, indicating that AOB can more effectively inhibit the oxidation of PC than that of PE. This may be due to the fact that AOB is a hydrophilic antioxidant, which can distribute more closely to PC than PE, because PC is more polar than PE, and then it can effectively locate itself at the oxidation reaction sites. The third part, summary. The oxidation of PUFAs being esterified into TAZ or PR all follow the first-order kinetic model. TAZ had a greater oxidation rate than PR, and PE had a greater oxidation rate than PC. PR could better be protected by the polar antioxidant than TAZ, and PC could more effectively be protected by polar antioxidant than PE. Monitoring oxidative substrate loss in conjunction with kinetic models can be used to compare the oxidative susceptibility of different lipid classes in complex foods. That's all. Thank you for your attention. Thank you.